Assalamualaikum warahmatullahi wabarakatuh. In the microbiology lesson, we are speaking about the laboratory techniques. In the last class, up to pasteurization, I have completed it. Now, following pasteurization, we are coming to the next heading, that is boiling. In the case of boiling, boiling of water. In boiling water, we place the materials for sterilization. In that case, the temperature is going to be 100 degrees Celsius. 100 degrees Celsius is a sufficient temperature to kill the pathogenic microorganisms. Most of the pathogenic microorganisms will be getting killed at 100 degrees Celsius. Saprophytic microorganisms, they are spores, mainly microbial spores. They are resistant to 100 degrees Celsius. Archaebacteria, like certain microorganisms, can survive above 100 degrees Celsius. That is one category. And second thing, it is spores. Microbial spores can withstand up to 121 degrees Celsius. That's why under moist heat method, we are providing 121 degrees Celsius as a standard temperature. So point it is, but boiling, okay, boiling is killing pathogenic microorganisms, most of the pathogenic microorganisms. So as an overall protection method, we can try boiling. After completing boiling, we come for filtration. There are special filters for this. And normally filtering mean you have some knowledge, you can, okay, size wise separation. Now, if you want to filter out bacteria, there are bacteria filters. If you want to filter out virus, there are virus filters. Okay, so filtering is one method, but bacterial filter cannot filter virus. Virus is comparatively very, very small. So bacterial filter has to work on bacteria or anything bigger than bacteria. Okay. So normally that is the filter we try. In case we suspect that is virus should be filtered only, we use virus filter. Why filtration? What are the requirements for filtration? To filter, that material should be in a liquid form or a solution form. Solid materials cannot be filtered. So anything that can be obtained in liquid or solution form can be filtered. Second thing, it is they are heat sensitive materials. If you heat them, if you try moist heat method or boiling method, due to that high temperature, they will be denatured. They will be they will be losing their functional qualities. So due to those reasons, moist heat method, like heating methods, are not applicable to these materials. In such a situation, we choose filtration. Okay, pause. Hope you can understand that one. Here you are mentioned what are the materials that are getting filtered. That you can see the list there. Thing it is, passing through this kind of tiny filter will be taking a longer time. It's not easy. It's not easy. It will be taking a very longer time for the filtration. To make it little faster, the filtering unit will be fixed to a vacuum pump. That vacuum pump will be slightly applied, so it will be sucking the liquid. So little by little, it will be pulling the liquid out. Then only it will be little faster. We can't apply too much of vacuum pull also, filter will be breaking. A very soft filter, it cannot be used with high level pressure. Right. So your book says in the filters there are various sizes there. 0.01 micrometer, 0.01 micrometer, 0.01 into 1000. If you multiply, it will be 10 nanometer, correct? 0.01 into 1000, it will be 10 nanometer. So 10 nanometer to 0.45 micrometer. That range of filters are there. I told you some of them are for virus, some of them are for prions. Okay. Okay, prions can be filtered, bacteria can be filtered, so various components can be filtered. Clear. Thereafter, we come for the next method, that is radiation. Especially, we apply UV radiation. UV, ultraviolet, can cause denaturation or damage to DNA. So when you apply UV radiation on microorganisms, their DNA will be denatured. Thereafter, they cannot perform their metabolism properly, so they will be killed. That is the method. Right? UV, your book says it cannot penetrate solid surfaces and covering, okay, and covering. So they cannot penetrate. Penetrating ability is very poor. So like we pointed on the moist heat method, in moist heat method, we told when we apply moist heat method to sterilize glasswares, each and every surface of the glassware should be exposed to steam. Otherwise, steam cannot completely sterilize it because steam cannot penetrate the glass. Same idea here. UV cannot penetrate, so each and every surface of that material should be directly exposed to UV. Then only it will be a perfect sterilization. But that is for paper, glass, okay, textile-like materials. Normally, UV radiation we try in the operation theaters, hospital rooms. So hospital rooms, operation theaters, you know, plenty of people come to operation theater. Different germs are coming to the operation theater. So these individuals should be harboring large number of microorganisms. So every time the operation theater to be kept very clean. Due to this reason, at a particular frequency, the operation theaters are applied with UV radiation, okay, to sterilize it. Okay, fine. No need to go very deeply about it. Nurseries, there is plant nurseries. Plant nurseries are plant nurseries. Okay, plant nurseries where the kuti plants are grown in large number. They can be infected by pathogenic microorganisms. So in such places also we use UV radiation to sterilize those places. So up to this one, if they are physical methods, now we are coming for the chemical methods. In the case of chemical method of sterilization, as the name it implies, chemicals are applied. There are many chemicals there. Here they are mainly pointing to gases. One it is ethylene oxide. Other one it is chlorine dioxide. The two major sterilizing agents. We use chlorine also, but now at the moment these two. Right? Majority of the chemical agents, your book says, majority of the chemical agents reduce the microbial population to a safe level or remove the vegetative forms of the pathogen. So this is an overall idea, right? Microbial population will be decreased, vegetative forms will be removed. Among these, ethylene oxide, if you consider, it will kill the microorganisms and their endospores also. It is also highly penetrating. Okay, so due to this reason, ethylene oxide is used to sterilize the mattresses in hospitals. Chlorine dioxide, another one, is a fumigate. Fumigation means it will be coming like a smoke. Okay, it will be coming like a smoke. So chlorine dioxide, okay, we fumigate. Now sambrani, that is a fume coming, right? Sambrani, smoke, fume. Actually, that's called fume. It's not called smoke. Okay, we can spread that one. Okay, here you are pointed, it is applied against Bacillus anthracis. Anthrax causing very powerful bacteria, one of the biological weapon, Bacillus anthracis, okay, is also considered. Chlorination, we are actually using calcium oxychloride. Calcium oxychloride for that, but people used to call it chlorine. Chlorine is a gas, you know, the bleaching powder we are using. Okay, the chlorination is also a sterilization technique. So up with this one, sterilization techniques are over. Now we are coming for a heading called preparation of culture media. Right? Microorganisms are cultured in the laboratories for various reasons. One reason, study purpose. Now in the university, we had a separate microbiology laboratory. There we have cultured various microorganisms. We have cultured them. Okay, why? For study purpose. Okay, some people will culture microorganisms for industrial purpose. For example, if you want to do a yogurt business, okay, you need Lactobacillus bulgaricus bacteria. From where you can get the Lactobacillus bulgaricus bacteria in pure form? You can go and buy in the shop. How? Shop is selling Lactobacillus bulgaricus. Yes, it's called by the name culture packet. If you buy a culture packet, in the culture packet there is Lactobacillus bulgaricus in large number. So point it is, culturing is very important. Bacteria. Naturally available microorganisms are very much diluted. Naturally available microorganisms are naturally available ones are very much diluted. In their diluted form, it's not easy to study them. So we are growing them. We are concentrating them in a place. Okay, so your book says natural habitat microorganisms you can't study. So we have to bring them to the laboratory and provide them similar conditions to support their growth and reproduction. To apply this, we are making a culture medium. Okay, to grow them, we want a culture medium, microbial culture medium. Right? In the case of microbial culture medium, it will be a jelly-like material, microbial culture medium. Right? I'll choose this one. You can see here a jelly-like material is there. It can be added to a petri dish or a test tube or a conical flask. That is not a matter. That jelly-like medium is the culture medium. Okay. When you go for the other diagrams here, when you look for the other diagrams here, in the other diagrams, you would have seen bacteria have grown on it. See, these are the bacteria grown on the surface of the culture medium. That's what you can see here. Okay, bacteria have grown. So bacteria will grow, fungus will grow. Okay, you can just search for fungus culture. Fungus will grow like this. Can you see? On the surface of the culture medium, fungus will be growing, bacteria will be growing. So looking at the fungus, their color, their colony shape, and we can make so many decisions about them. Okay, right. So this is how the culture medium. Okay, so culture medium, what is culture medium? It's a nutrient material prepared for providing nutrition and encourage essential to growth of microorganisms at laboratory conditions. nutrient material prepared for providing nutrition and encourage essential to the growth of microorganisms at laboratory conditions is called a culture medium. Another point you are given, not all microorganisms can be grown on a laboratory culture medium. For an example, uh, phytoplasma cannot be grown on culture medium. You already studied that one. Phytoplasma cannot be grown on culture medium. So we can't expect all the ones can be grown on culture medium. Similarly, virus cannot be grown because virus need a live medium. That means we used to grow virus on a chick embryo. On a chick embryo, we grow. Why? Because virus is an obligate parasite. Okay, virus is a obligate parasite, so it cannot be grown in a culture medium. If you give a live culture, it will grow. Understood? So, some microorganisms grow well on any culture medium. Some microorganisms require special culture medium. See, variations are there. Some microorganisms cannot grow on any culture medium. Opposite, can grow. On the can grow ones, another two types are there. One, any culture medium, okay. There is one category. Special culture medium are essential is the other. Okay, right. In a culture medium, sufficient nutrients should be there. Moisture, sufficient amount should be there. Suitable pH should be there. All these things we have to maintain. Right? When we prepare a culture medium, after preparing the culture medium, we are the one who introduce the initial sample from what the microorganism has to grow. For an example, if you want to find out the microorganisms in a soil sample, it is you or we need to take the soil sample and spread on the culture medium. Then that microorganism will be growing in that culture medium from that soil sample. If you want to find the microorganisms on your hand, okay, if you want to microorganisms on hand, I have seen a picture. Now see this one. One person has kept his palm on the culture medium. Okay, so what has happened is the microorganisms have in his hand have grown there. So see how nice it is. You nicely looking it. Right. So that is the idea. So it is you going to introduce it. In that case, before you introduce, can there be any microorganism there? Mean no. There cannot be any microorganism. So, so okay, if there are any other microorganism, what would happen? They also will grow. Then which one is coming from your source, which one was existing there, we are not clear. To that reason, what we do is, first of all, after completing it, we will sterilize that medium. Okay, the glasswares using there should be sterilized. The materials used there should be sterilized. After sterilization, you have to introduce your sample. Then only your sample microorganisms only will be received. Understood? Good. And thereafter, we are coming to another heading. Another heading means another point. In the culture medium, there are two major type of culture medium. Actually, there are certain other types of culture medium, but two major types are these two. One it is nutrient agar, other one is potato dextrose agar. NA and PDA, they are standards. Okay, you can go to a laboratory shop and ask them to give you 10 grams of PDA. Then they will ask you to pay 12,000 rupees or 25,000 rupees for 10 gram PDA. That much an expensive material. But you can prepare. That is something simple. But people go for ready-made things. So ready-made things are very, very expensive. So there are two media. Nutrient agar, other one is potato dextrose agar. Nutrient agar, if you consider, we are making it using peptone. It's a protein source, a yellow color protein source called peptone. I will show you peptone. So this is peptone. Okay, peptone powder. Actually, it will be, it's a bad smelling one. I have seen that one. I have smelled it also. Okay, off white. Yeah, yellow color mean yellow dhamma. So I have seen yellow one. This is looking like an off white one. Yeah, off white. Oh, see here. This is what I have seen. Yellow peptone powder, a bad smelling one. It's a protein source. Okay, we are using peptone, the protein source there. That's in the laboratory. Meat extract by boiling some pieces of beef and taking that extract, we use it. Sodium chloride. Sodium chloride is very essential for the bacterial growth, so we use it. Agar is a solidifying medium. Agar has no more role play. It will make that medium solid. And distilled water. On the other hand, PDA mean potato pieces, they are extract, glucose, those things we use. Nutrient agar rich in protein. Because it is rich in protein, nutrient agar is good for bacterial growth. PDA is rich in carbohydrates, so it is good for fungal growth. Okay, right. And next it is Agar, I told you, is already a solidifying medium. It makes that medium solid. If it is solidified only, the bacteria will be growing on the surface of the medium. Otherwise, the bacteria will be penetrating through the entire medium. So solid medium are very important for us. We want the bacteria or fungus to grow on the surface, then only we can observe them. So agar is not a nutrient for the microorganism. Agar is not a nutrient for the microorganism. Due to that reason, we use agar as a solidifying medium. Okay, when you get agar, it will be also an off-white color or a yellow color powder. When you heat it above 40 degrees Celsius, little by little, it will dissolve in water. Later, if you allow it to settle, it will be becoming a solid medium, right? So solid cultures are possible using agar, right? So further, because most of the microorganisms appear almost colorless when viewed through a standard light microscope, we have to prepare them for observation. That is a separate story. We will speak about that story in the coming one. I have to go to school now. So I am finishing the portion with this. Okay. Assalamu alaikum wa rahmatullahi wa barakatuh.